Hey guys, so this is going to be about an important topic from genetics for biochemistry. So this is about the DNA replication. Uh, Before I start about DNA replication, I just want to know. I just want to tell you that after DNA replication, I'll talk about recombinant DNA. So recombinant DNA form like the process of forming that is gene genetic engineering, and rDNA is chimeric DNA. Now rDNA is called as chimeric DNA, and this has been asked as an MCQ very often, so I've just mentioned that point over here itself. Then we move on to actual DNA. So introduction of DNA as the structural component was taught in proteins itself. Like it is initially taught to us in proteins, and even in 12th we have learned uh, about the basic nature of the double helical structure and the complementary base pairing and the anti-parallel nature of the DNA. So that is not something I'm going to go through. I'm just going to go through the DNA replication. Now it is an important part of the central dogma, which is basically the process of conversion of DNA to proteins, right? So first DNA gets converted into RNA via transcription, and then translation converts this RNA into proteins. And DNA replication is the first step of central dogma, where DNA forms another DNA. So a parent DNA is going to form a template DNA. Now moving on to the nature of DNA replication, which was semi-conservative, which was confirmed by Mendelssohn and Stahl in the experiment they did with heavy nitrogen using nitrogen 15, and we have learned this about in 12th, so I'm not going to go through with it. But the only thing that they did was they introduced a heavy isotope or a different isotope of nitrogen in one of the DNAs, and as replication went on, the content of that heavy nitrogen helped them to determine that the nature of DNA replication was semi-conservative, which meant that only one parent strand took part in the formation of the next DNA. So like let's suppose one DNA having two strands is taking part in DNA replication. The two DNAs that are going to be getting formed after the replication will have 50% of the parental DNA, right? So after that we move on to the actual process. Now this is going to be quick. What I'll go through is it in prokaryotes, and I'll just list the differences that are seen in eukaryotes. Now first studied in E. coli, and requirements, like this is the way I'll usually go for attempting an LAQ question, because DNA replication is an LAQ question. So you go for the basics, like give an introduction that you are mentioning about the DNA replication in prokaryotes, then go through with the requirements of replication, which is basically all the nucleotides, D ATP, D GTP, D CTP, right? Then we can go through with RNA primer, then DNA helicase, then single strand binding proteins, then DNA polymerase, DNA ligase, and topoisomerases. Now all these are required in various steps, which I'll list when we go further. Then we've got three types of DNA polymerases, essentially DNA P1, that is polymerase 1, 2 and 3. Now they are asked as a 2 marks differentiation between. So the main function of DNA 1 is going to be gap filling. Now what gap filling means is that when nucleotides are basically getting joined, if for some reason there is a gap between two nucleotides, then that is connected via the DNA polymerase 1. DNA polymerase 2 is going to take part in DNA repair. DNA repair and damage is a very important question for 5 marks, which I'll cover in the next video itself. Then DNA polymerase 3, that is replication of that is going to take place part in the replication of the leading strand. Now leading and lagging strand, as we know that leading strand is basically a continuous array of DNA getting formed, while lagging strand is basically Okazaki fragments, which are going to get formed bits by bits, and they are going to get connected via the DNA ligase enzyme. And only DNA polymerase 1 has 5-3- exonuclease activity, which basically means कि जो भी primer हम insert करने वाले हैं, the RNA primer that has been inserted before replication, that can be removed by exonuclease, क्योंकि 3-5- exonuclease activity is present in DNA 1, 2 as well as 3. ओके और सबसे ज़्यादा ये नंबर्स याद रखने की ज़रूरत तो नहीं है वैसे, बट इफ़ you remember it is well and good, but you should know that the maximum rate of nucleotide formation per second is by DNA polymerase 3. That is why it is the main polymerase that is going to help in DNA replication. Moving on to the process of DNA replication. So now the process basically has 3 steps, which is initiation, elongation and termination. Initiation is going to take the various steps of initiation are as follows. First, ORI fragment is recognized or established. That is, ORI is origin of replication, which is just a series of 245 base pairs, which is densely which is majorly adenine thymine base pairs. Now the reason is very simple, that the if you want to disturb or if you want to break the bonds between two base pairs, it better be less number of bonds. So AT bond is just a double hydrogen bond, so it is easily it can be easily broken up. Then we go ahead and we see that 2 bonds are easier to break, and this leads to the formation of a replication bubble. Now each replication bubble is having 2 replication forks, as we can see. Formation of replication fork is by a DNA helicase. Helicase is basically just going to pull the 2 strands apart. So think of it in this way, that if somebody asks you to cut like a plastic piece from the center, not from the a plastic square piece, not from the edges but rather from the center, what you'll do is you'll take the a pointed thing, you'll poke a hole in it first, then you'll take the scissor and then you'll start cutting from the hole, right? So that is what is taking place. First what happens is DNA A is at first there is going to be the formation of a replication bubble, which is only the local separation. So only local separation is going to happen there with the help of DNA A protein, which is just like the hole, and then helicase will completely separate the strands, which will be like extending it with the scissors. So that is what is going to take place in the first step. So each replication bubble has 2 replication forks. This is need to can be asked as a MCQ. Now stabilizing of the DNA is done via the ssDNAP, that is DNA proteins, which is single strand binding proteins, which help the single stranded DNA to be intact, which is basically avoiding the reformation of the hydrogen bonds, because DNA has an innate tendency to get repaired. Then we have releasing the supercoils via topoisomerases. Now what is supercoiling is basically the 2 strands can kind of get the separated strands can get knotted. Now to remove the knot, what the topoisomerase is going to do is it is going to form a nick. What is a nick? It is going to cut this portion of that knotting. So basically this X will be removed. Then we'll get 2 strands separated ssDNA. Now what will happen is there is a scope for the rotation of these strands, because A needs to get paired with the ssDNA strand, and B needs to get paired with the B DNA strand, because we see that because of the knotting the strands have rotated to a different location. So now they rotate at first and then they again get joined to form the normal DNA without any knots. So this has been asked previously, what is nick formation. So this is something to remember. Moving on to RNA primer addition and primosome. Now what happens is, in case of leading strand, only one RNA primer is required, because leading strand is going to be continuously formed, because 5-3- is the direction of you want to have for the formation of DNA, 5- to 3- end. So that is present in leading strand. So you will only require one DNA primase, RNA primase, and for lagging strand you'll require more than one RNA primer. So what happens is, on the lagging strand, when this primase, like how are these primers exactly put in? So it is via the structure called as primosome, which is basically primase along with some single stranded binding proteins with some other proteins. This will form this structure called as primosome, which will move along the lagging strand from 5' to 3' end and start putting few RNA primers along its way wherever it is necessary. So then we have got elongation. Elongation, the direction should always be from 5' end to 3' end. So simultaneously on the both strands the elongation is going to take place. Now proofreading, or basically what you need to understand is the enzyme, that is DNA polymerase 3, can read the DNA from 3' to 5' end. So it can synthesize the DNA exactly opposite, क्योंकि complementary pairing होने वाला है. So anti-parallel pairs are actually formed. So if you are if the DNA polymerase is reading from 3' to 5', it will have to form the DNA from 5' to 3'. So that is how DNA pairing is going to take place. Now this 3' and 5' is basically what part are we referring to, right? 5' is the phosphate part of the DNA, or the hydroxyl part is the 3'. So this is something which is actually in the structural component of DNA. If you want a video, please let me know in the comments. Then leading strand, 3' end of primer acceptor of first deoxyribonucleotide, and then, like for example, if this is the parental strand, then from 3' to 5' end will be the parental strand, while 5' to 3' will be the it will be the new DNA. And then as it is moving forward, phosphodiester linkages will be formed between the various parts. Now important is lagging strand, where Okazaki fragments are formed, since the direction of formation, as I told you, is opposite, and you need 5' to 3' end, but it is actually 3' to 5'. So that is why we need the primers at every step. Then for termination we have got a terminus sequence of 20 base pairs, which is when the replication fork reaches that segment, then the DNA replication gets terminated. Now we move on to the eukaryotic DNA replication. Eukaryotic DNA replication, I've only mentioned the differences because the rest everything is almost the same. Now the differences is basically, first and most important, is DNA polymerase is not 1, 2 and 3. We've got 5 types, which range from alpha, beta, gamma, delta and eta. So these are the 5 types of, in which for alpha, if we just have a look for alpha, so for alpha we can see that we have got primase activity. For beta we have got repair activity. For gamma we've got mitochondrial DNA it is present in the mitochondrial DNA replication. Delta acts like DNA polymerase 3, which just means कि जो भी leading और lagging strand का formation most of the होगा DNA का, वो डेल्टा करेगा. यानी कि सबसे इम्पॉर्टेंट जो डीएनए पॉलीमरेज है एक तरीके से वो ये हो जाता है, और ये भी हो जाता है बी, क्योंकि यूकैरियोटिक डीएनए इज मोर प्रोन टू डैमेज, एंड दैट इज वाई फॉर रिपेयर यू नीड डीएनए बीटा. सो दैट इज वाई दीज टू आर द मेजर डीएनए पॉलीमरेज इज रिक्वायर्ड. देन वी मूव ऑन टू द initiation, which the difference first being that more than one origin of replication is going to be present, not only one, which was only one origin of replication in case of prokaryotic DNA. Then gap of 50,000 to 1 lakh base pairs can be seen between each origin of replication. Then elongation. For elongation, a clamp is required. Even in prokaryotic DNA clamp is formed, but what happens is in this the slider is also present. You need to slide the DNA polymerase from one part to another, and it will just keep binding the nucleotide. So you need a sliding part also, which is which is basically the proliferating cell nuclear antigen, which is pCNA. So that is important. That is first we'll get the replication factor C binding to the origin of replication, which is the clamp. Then we need a slider for that clamp. That slider is going to be pCNA, which is going to slide the clamp along. And what will happen is that DNA polymerase delta will just get adhered to the clamp first, and then this clamp, with the help of the slider, will keep moving. The DNA polymerase is on it, and it will keep binding the nucleotides as it moves along the DNA. So that is what basically occurs. Then we've got termination. Termination is via the end of the linear chromosomes, which is called as telomerases. Now telomeres themselves are asked as a 5-mark question sometime. 5 marks or 2 marks, but it is very important because it is literally asked every year. Even if it is just an MCQ, they had asked the sequence, right? So you can also remember the sequence, which is TTA-GGG, and just this sequence is repeated in the telomere. Now Hookston hydrogen base pairing is present. Now why is this important? Hookston hydrogen pairing is simply guanine rich pairing, like this means guanine rich pairing. So that is why you need to remember the type of pairing, which had been asked once as an MCQ many years back, but can be asked. So I've just mentioned it. If we'll go through with the telomerase, which is the enzyme present in this region. Telomerase is basically going to add the telomere. Now what adding of telomere does is it is going to terminate your DNA replication. So if you want a certain length of DNA to be formed, you just attach the telomere to this end, and only this much DNA will be replicated. So it is kind of a fail-safe mechanism for how much DNA you need to form. If uncontrolled DNA is getting formed, that can lead to cancer, right? So in cancer we'll see increased telomerase activity. Then some salient features quickly. Repetitive DNA sequences which are found at the end of the chromosome, right? Then 46 chromosomes are present, right? So 46 chromosome, हर एक chromosome के दो end होंगे, एक upper end और एक lower end. So we've got 92 telomeres basically. Then it acts as a capping of the shoelace, like shoelace के end पे जो वो plastic का लगाया हुआ होता है, which is going to actually bind the shoelace together, right? So it cannot unwind. अगर आप shoelace को उल्टा सिर्फ घुमा दोगे तो it will unwind, right? So to prevent that, we've got that cap at the end, which just prevents the unwinding, which is like the action of telomere. Finally, moving on to the last part, we've got provides a buffer zone where chromosomes can get shorter but no DNA is lost. What does this mean? कि the chromosome gets shorter but no DNA is lost. It basically means कि usually DNA अगर हमको शॉर्ट फॉर्म ही करना है तो उसके आगे का एक पोर्शन जो है वो लाइगेट किया जाता है, उसको कट किया जाता है ना, using DNA like exonucleotidases or anything, right? So you cut the DNA, but in that case DNA is lost. What you can simply do is आप telomerase की एक्टिविटी बढ़ा दो और वो जो लेंथ है टीलोमियर का उसको बढ़ा दो, तो DNA तो वहीं पे रहेगा बट उसके आगे मतलब DNA फॉर रेप्लिकेशन नहीं होगा, तो दैट DNA इज कंजर्व्ड. बट बिकॉज वहाँ पे टीलोमियर है तो वो DNA रेप्लिकेशन में पार्ट नहीं लेगा, सो DNA इज कंजर्व्ड एज वेल एज रेप्लिकेशन इज टेकिंग प्लेस. So it is acting as a buffer zone, right? So role of telomerase in degenerative diseases, कैंसर में उसकी एक्टिविटी बढ़ जाती है. और प्रोजेरिया एक कंडीशन है जिसके अंदर टीलोमियर छोटा हो जाता है बहुत. अब छोटा टीलोमियर होगा तो जो सेल प्रोलिफरेशन होगा वो अफेक्ट हो जाएगा, उसकी वजह से एजिंग वगैरह दिखना स्टार्ट हो जाता है, राइट? सो like the aging process is basically accelerated. Now we go on to the inhibitors of DNA replication. This is probably the last part of DNA replication, and I've covered everything, so you can write everything as a long question, like if they ask you for the long answer question. So this is the final thing you need to write, which is basically what are the drugs that inhibit DNA replication. So the nature of the drugs and the list of the drugs. So antimicrobial drugs can be used. Antimicrobial drugs, I'll just see the list, which is antimicrobial drugs like novabiocin or ciprofloxacin. Ciprofloxacin has been asked before as like what is the function of ciprofloxacin. So it is an antimicrobial DNA replication. Like anti-cancer proper drugs, we've got actinomycin or doxorubicin. And then we have got nucleoside analogs like 6 mercaptopurine and 5 fluorouracil. AZT is also used. AZTs are also like AZT is the most used, I'll say, latest drug, I guess, because whenever you search anti-cancer drugs on the internet, the first drug usually that pops up is either mercaptopurine or AZT, because AZT is a very latest form of treatment which actually like acts against the polymerases, DNA polymerases. So what is cancer is basically more DNA formation and more cell formation. But if DNA polymerase activity is inhibited, you can easily guess कि DNA replication जो होगा वो कम हो जाएगा. So that helps to reduce the cancerous cells proliferation. So that is why it has been used widely. So you can go through with these examples, and I hope you liked the video. If you did, please hit the like button and subscribe if you and if you need any topic to be covered further on, please let me know in the comments. Thank you.